Good morning, everybody. Welcome to today's teaching series. I'm going to do the introductions and absence of Lent. So today we will get a talk from Dr. Stephen Till Tillis, thank you, who is a clinical professor at the University of Washington. And I think, as many of you know, he has previously worked in private practice and has done a lot of clinical research there, as well as in industry. And he'll be talking to us about food allergy treatments. So looking forward to it. Thank you.
Thank you. And hopefully everybody can hear okay. And thanks for having me. It's a pleasure to come back again this year. And hopefully will not be overly redundant to what we talked about last year. And I was asked to talk primarily about things that are kind of new. And I'm going to make sure I'm not sure we're recording or not. But anyway, we'll just get on. Okay, good. All right, perfect. So food allergy treatments 1 year on was my assigned topic. And let me talk a little bit. My disclosures, first of all, as Anya mentioned, I was in private practice and helped run a research center in Seattle for 18 years, and then joined Immune Therapeutics a little over 5 years ago, almost 6 years ago, actually, and then retired from that a year ago. So that's still relevant because they're the manufacturer of Palforzia or PETA, which we'll talk about today. Right now, I'm a consultant with FAIR and also Allergens Greer, which is now the owner of Palforzia. So I still am advising them about a strategy, and also have a couple of other consultant relationships. The objectives today are a little bit different than because there's only 1 hour, and this is becoming an increasingly complex topic, which is good. Food allergy needs more stuff. But we'd like to understand the safety and efficacy data for the FDA-approved treatments, appreciate other factors relevant to food allergy shared decision-making, which is something that is now in all of our, as prescribers, all of our everyday lives. We sort of have a responsibility to do responsible shared decision making, but also appreciate the pros and cons of standardized allergens, which has become a thing when you're using them for immunotherapy for food. And as an outline, we'll start with a sort of unorthodox background that'll be as brief as possible, then review FDA-approved treatment data as a review, because this is mainly what I talked about last year, but it'll be much shorter. And then an update, not an update, but put in context the epicutaneous immunotherapy Viscin peanut data, and then stuff that's more relevant in the past year. But let's start with just a reminder about when you interpret a published manuscript, especially when there's clinical trials or patient data involved, there's a whole range of certainty about the scientific evidence. And you know this, but there's these grade recommendations. And having been involved with the practice parameter in the past, it's a dilemma if you as a specialty confine yourself to only grade 1A, certainty evidence, then you have very little to say, because it's very hard to go beyond that, because it's so hard to do those kind of trials. And so, consequently, most of the stuff that we rely on is oops, I'm messing things up here. There we go. Most of what we rely on is D, which is expert opinion based on interpretation of evidence. And that is sometimes wrong. There's some misadventures in the past that include, for example, when I was trained, you were supposed to avoid peanut until age 3, for example. And I think I single-handedly caused peanut allergy in several hundred little kids that are now adults. And so I think we need to have a healthy skepticism when we're interpreting data. Remember, also a pivotal trial, which is a phase 3 trial, especially with efficacy, but generally efficacy and safety, is even a step above the randomized controlled trial because it's scrutinized by the FDA in a way that has a much different and more thorough underpinning. So just a reminder of that stuff, which you already know. in this slide, I'm going to try to very quickly go through some review of the past 25 years, which basically my career, a little bit shorter than my career, but is sort of what I observed in my own clinic. And on the bottom, the first two bubbles didn't arise. One of them was really small, and then the one that's blank there is a little bit bigger, as you can probably guess. But then the point is the unmet need has continued to grow in food allergy every year, and certainly every decade, and at least, I think conservatively, every 2 decades it doubles, but it may be quite a bit more than that. And looking at, there was a recognition of this at about 15 years ago or so. And the Food Allergy Initiative, which was an advocacy group at the time, as later became known as FAIR, had a summit, and they concluded that there was a need for the pharmaceutical development of an oral immunotherapy for peanut, because there had started to be evidence in that area. And so they actually triggered the formation of a company called ARC, which was later became Immune. And so the former CEO of Immune used to say to people that it was the first crowdsourced biopharma company. Anyway, so that was a long time, not a long time ago. I guess it depends how old you are, how long ago that was. But this is how this happened. There was no big pharma company that was willing to do this at that time. Later, the FDA advisory committee sort of concluded, or the FDA decided based on recommendations, that double-blind placebo-controlled food challenge would be the outcome for efficacy that would have to be used, which has proven to be interesting, and also a barrier to high volume studies, because it's so tedious to do, as many of you who've been involved in these trials know. And then about the same time, the Academy did a survey that said, based on data about oral immunotherapy, 70% of respondents said, yeah, I would integrate this into my practice, but only if it was FDA approved. And it turns out that's not what happened, but that was the state as of about 2015. Then phase 3 peanut OIT was initiated. It was approved in age 20 for ages 4 to 17, or in 2020 for ages 4 to 17, and then age 1 to 3 last year. Now, going even further back, the anti-IgE concept, it wasn't, it's not new. This was way back before the year 2000. There was thinking about this, and there was a New England Journal article, I think it was 2002 or something like that, that was a phase 2 study with an anti-IgE called Tanox 901, which is very similar to omalizumab, and had very promising results. But due to some legal shenanigans between manufacturers of anti-IgE molecules, they were put on hold, and that company was since acquired. But anyway, this was way back then. Then we were investigators in that trial, and then also this other trial in ligolizumab, which is a term familiar to all of us. In I think it was 2012 or something, that they had planned a phase 2 trial. We went to an investigator meeting, and then it was canceled shortly thereafter. So a long history. Meanwhile, there's all these controlled studies with omalizumab and peanut allergy, and omalizumab with OIT, that were very promising, but it didn't ever get developed. Finally, the phase 3, that was a sneak preview. The phase 3 trial for omalizumab was done, and it was done by NIAID, so the NIH, in cooperation with Genentech, who provided the drug and funding, but not the army of clinical development people that I now know, having been on the inside, are pretty impressive in terms of how a company can actually get something to market with very high quality evidence. And then finally, a year ago or so, was approved, and you all, I know, are very familiar with that. So, and then finally, you see the peanut phase 3 epicutaneous immunotherapy called Viaskin Peanut was initiated at the same month that the oral immunotherapy for peanut was initiated. And then we'll talk a little bit about what have the impediments to why that's not available yet. Anyway, so that's a little history. Here's another one that is, I think, really important to keep in mind. We don't have time to go through the whole study, but back in, I think it started in 2012, so a long time ago, they thought, okay, maybe toddlers would be a good patient group to try oral immunotherapy with peanut. And in fact, it was. And then in 2022, the study called Impact was published. This was done by the Immune Tolerance Network, which is also the network that did the LEAP study, as you're all familiar. So very high clinically relevant results that come out of this group. And briefly in blue or in red are the patients who you could predict remission in this subgroup based on two things: the patient's age and the patient's baseline specific IgE. So the lower the age and the lower the specific IgE, the more likely that they achieve remission, which was defined as the ability to tolerate 5,000 milligrams of peanut after being off all sources of peanut for 6 months. And this is after 2.5 years of OIT, then complete avoidance for 6 months, and then another double-blind challenge, tolerating 5,000. So a high bar. And you can see the older the patient, the less likely. But a lot of these toddlers, in fact, more than 70% of the toddlers that had started the treatment prior to their 3rd birthday were able to achieve remission in this study. So it's introduced a new type of concept in an age group that really we don't talk about as much with the older patients. All right, so whoops, this is a jumpy thingy here. So let's go back. Let's talk about FDA-approved treatments as a review. And remember, the pivotal trial's primary outcome is the one that we're most certain about, and then exploratory outcomes or secondary outcomes, and then exploratory outcomes. You have permission to think about if the primary outcome is convincing. And then, so it's called a hierarchy, which is really important. And these things are established prior to the start of the study, which is also really important. So let's talk about the FDA-approved thing. So the first is peanut arachis hypogea allergen powder DNFP. So that's an unattractive generic name for what is now known commercially as Palforzia. And so the abbreviation in the literature is PETA, which I don't like, but that's nevertheless what it is. And in the development program was called AR101. So all those names are the same, mean the same thing. And there are two pivotal efficacy and safety trials, one in ages 4 to 17 and one ages 1 to 3. And we'll look a little bit. This is sort of a side-by-side, not sort of, it is a side-by-side look. I won't say comparison because it's hard to compare when it wasn't the same trial, but the study designs were similar. And in a nutshell, so to speak, let's look at the primary outcome, which is in the middle on the top here, 600 milligrams in age 4 to 17, roughly 2/3 that achieve that in an intention to treat analysis compared to a very low level of the placebo responders in that group. And again, this is immunotherapy. They're taking it every single day. So there's no surprise that this would work. But the point is it's more than the maintenance dose, which is 300 milligrams. And you can see on the right, as a secondary outcome, half the patients tolerated 1,000 milligrams. And another semantic thing that's really important is the outcome description. So highest tolerated dose was the outcome here, 600 milligrams, and highest tolerated dose of 1,000 was half the patient. There's also terminology called eliciting dose, and that for the half of the patients that tolerated 1,000, we don't know the eliciting dose because the last dose of the challenge after 12 months was 1,000. So important to keep that in mind as you're looking at the results of studies. And in the 1 to 3 group, you can see very similar results, though the numeric proportions for each outcome were higher, including an exploratory outcome of 2,000 milligrams, where nearly 2/3 tolerated 2,000 milligrams after just 1 year. So this is 4 to 8 peanuts or 6 to 8 peanuts in a toddler who started out tolerating 1/10 of 1 peanut. So it's again showing what we all know as allergists, that with immunotherapy, you stay on the maintenance dose and it continues to protect beyond what the dose you're giving them as the immune system changes gear. This is exploratory outcome, which I would only do because the other outcomes were so strong, that's giving a little bit more sort of, low quality evidence, but at least it's interesting to look at, of what happens with the even younger ages. So you can see this is the same data but looking at ages 1 to 2, 2 to 3, 3 to 4 at the time they started treatment. And you can see, for example, the primary outcome, instead of 2/3, it was like 81, almost 82% in the age 1 to 2 group. And we won't go through all of these, but also notice that in the, and let's see if that's, oh, you're seeing my whole thing. Let me get rid of this because you're seeing that too. Yeah, there we go. So you're seeing on the far right that if the patients were 3 years old at the time they started the study, none of them achieved the primary outcome. So again, not high quality, but really supporting that hypothesis that younger is better. And we'll get to their threshold at baseline in a minute, because that's really important these days. Now, safety-wise, with the peanut or PETA, I think the main things are, about 11% in the 4 to 17 discontinued due to adverse events. And that's, I think, probably not news to any of you. In age 1 to 3, it was almost nearly half, but certainly a lower percentage for whatever that's worth. Gastrointestinal adverse events, I think of with OIT, because I'm an internist that did SCIT for a living for a couple decades before I even started working with food allergy. It's kind of like SCIT in that local reactions with SCIT are bumps on the arm. Local reactions with OIT are belly pain or various sort of not very severe forms of GI symptoms that occur acutely. And then there's some other adverse events of special interest, one of which is unique to OIT compared to SCIT, which is GI adverse events of special interest, which, for example, would lead to discontinuation of the product or possibly EOE. It was 6.5% who discontinued in the age 4 to 17, and 3.1% who discontinued due to GI adverse events in the toddlers. And then finally, systemic allergic reactions, which in the label for, and I think in an abundance of caution, the FDA refers to what we as allergists call systemic reactions, anaphylactic reactions. And then they grade it mild, moderate, or severe. So it's like, mild anaphylaxis, if you will. And anyway, so it was a 14.2% in the 4 to 17, 8.1% in age 1 to 3. And in that pivotal trial, the placebo had the same, this is all-cause systemic reactions, were no different. And then there were no severe reactions in the toddler group and very few in the older children. And then leading to discontinuation was rare. So sort of like with SCIT, where there are patients who have to discontinue, but it isn't necessarily the beginning of the end for most patients. And then epi use, which I'll circle back to, because I mean, trying to interpret severity or risk in your patient, epi is a very interesting and confusing endpoint or marker, because we tell patients and parents to use it if they are worried. So it kind of reflects their degree of worry at that moment, but it doesn't necessarily reflect the severity of the reaction or anaphylaxis, although it could be important because it could prevent severe reactions. We just don't know the answer to any of those questions. So in all of these studies, which we'll go through, there is noise in epi. Exactly. This is not oral challenge associated. This is during the year. These are all low threshold patients, most of whom had other allergic diseases and other foods that they're allergic to. So kind of a noisy background, and you could see and that's reflected in the placebo rates of epi use. And I'll also highlight this with omalizumab and also the Viascan, because it's actually very similar in the different studies. Okay, so I won't belabor that more. I won't spend a lot of time on this slide, but just to say that with PETA, it's been on the market now for 5 years, and so the clinical development program followed those patients that were in the development program for over long term. And you can just see, as expected, the most of the adverse events are in the first year or so, and then there's many fewer. And in light blue is the treatment related. Definitely ask yourself, this isn't, remember those grade evidence things? This is open label follow-up, and there's survival bias, all kinds of things that could be used to criticize this. What I would point out is that remember, there were 11% who discontinued in the 4 to 17 due to adverse events. If you look at the entire development program, how many discontinued due to adverse events over even the long-term extension, it was 13%. So it wasn't that many more who discontinued due to adverse events. There are a lot of patients, in fact, the majority, who discontinued because they switched to grocery store sources of peanut once they were on maintenance. So it's interesting. But anyway, I think it's keep all that in context. You know, if we inflate the y-axis, so this is exposure adjusted, and as a comparator, this previous one, it goes on a scale from 0 to 75.
Hi.
Hello. All right, so hopefully that's okay. If you go from 0 to 0.3, you can actually see some discrimination of anaphylactic reactions and epi-use in these patients. And it takes probably until, or it seems to take between the first and second year, or partway through the second year, before I'd be convinced that there's any decrease going on here. But it seems to trend down after that, which is, again, not surprising. The most common reason for and severe reactions haven't actually occurred after year 2 so far in those groups. And the most common reason for more severe reactions is patients exercising too soon after a dose. All right, well, let's change gears. That was a very quick overview of the PETA group. So let's look at omalizumab. You remember this from a year ago, the New England Journal study, stage 1, which was the pivotal trial. The rest of Outmatch was not a pivotal trial, but the stage 1 was. And this, the primary endpoint was after 16 weeks of omalizumab, would they tolerate 600 milligrams of peanut? So all the patients were allergic to peanut. The 600 milligrams of tolerated it's not coincidence. That was very similar to the study design of the other pivotal trial with PETA. So, and shockingly, the 67 or 2/3 or so is a recurring theme in all of these studies. So 2/3 of them in an intention to treat analysis achieved that 600 milligram threshold. And you can see what's more exciting is that a bunch of other foods had statistical significance with these key secondary endpoints. Cashew, there was a little blip, which we won't get into, but relative to placebo, this seemed like a pretty compelling validation of what we already knew from a lot of studies over the years. I'm not even going to talk about safety data because it's been on the market more than 2 decades, and there weren't any red flags in that data. So pretty impressive. However, and their job is to be critical of things. So you can see here when there's looking carefully at the peanut, which was the primary outcome, you can see that black line is a nice depiction of how the group increased. And with clinical studies, you get outliers that do all kinds of weird things. In this one, and the efficacy was on average excellent. However, the 14% of them couldn't even tolerate more than 1/10 of one peanut, which is so it reminds me of with omalizumab and chronic urticaria, where there are some patients that don't seem to respond well, and we don't know why. This is the clinical trial. It's a pivotal trial, so it's really high quality. However, the real world, it might be better. I don't know. I mean, I haven't heard things in terms of real world anecdotal evidence that these non-responders are having problems. But it's important to keep this in mind, that it was different than OIT, where if they're tolerating it, there's a partial protection, whereas it seems like some of these patients didn't have that for whatever reason. And then finally, as we'll get to in a little while, the potential for combination with OIT is obvious. That had already been published, and we call that an adjuvant usage, and we'll come back to that because that was just presented. It's not been published yet. Now, I promised a little update on, not update, but just sort of acknowledgement and circle back with the theme of today, which is quality of evidence. So the pivotal trial primary outcomes in the Viaskin studies were in 4 to 11 and ages 1 to 3 was the eliciting dose. So again, the dose that causes symptoms, not the dose that they last tolerated. That the eliciting dose would be 1,000 milligrams or more. And you know, there was another way to get there, but this is the vast majority of patients were in that category. And you can see on the left that there was statistical significance with the difference between treated and untreated. And it's interesting that they chose to add all these zeros in the p-value. Normally, the journals won't let you add more than 2 zeros typically, but anyway, there's only basically 1 out of 3 patients that responded in that study. Somebody's not muted, but that's okay. At least there's people out there. Anyway, so it technically was a negative study because the lower bound of the confidence interval wasn't great enough. And to cut to the chase, in phase 2, because we were investigators, or we did that in Seattle, and phase 2, it went all the way up to age 17, but it didn't work in teenagers, so they went from 4 to 11. So spoiler alert, it works better in 4 to 7, which is what now they're developing, which is great. And then in the age 1 to 3 on the right, which is also a pivotal trial, they did achieve their statistical significance, and the confidence interval, as you can see, was 22.4, the lower bound, which exceeds the minimum of 15%, which is what you need in order to call it a positive study. Interestingly, a third of them, of the placebo patients, also achieved the primary outcome. So, but I think that puts in perspective why it's taken so long to get approved, that even with, and then there are some patch things about adhesivity and how regularly it gets absorbed into the skin. But with any luck, it'll get approved in the next 2 years or so. They still have to do another pivotal trial in age 1 to 3. In age 4 to 7, they don't have to do another trial, but they have to analyze the data, and they've said that they're going to submit a year from now or so. And then it takes a year to get approval. So in a couple of years, I think you can maybe have yet another option that's FDA approved. They've been talking about it, has not been initiated to my knowledge. I can't find it on clinicaltrials.gov. Yeah, so it's interesting, and it's unfortunate because the patients have been waiting for this for 15 years. In fact, you were a fellow, if I remember, when we were doing oral challenges on these patients. Anyway, so, and looking at Viaskin safety, I mean, everybody knows it's really safe, right? And I think that's true, but it's the devil's in the details, I think. And I think just to illustrate the noise that are in these pivotal trials, let's look at so there's not a lot of patients discontinuing. The GI adverse events, I don't know what that's all about. I don't believe that there's legit GI stuff that's happening because of the patch. And that's reflected in the 1 to 3 here, where placebo had just as much. Systemic adverse events of any cause, which is really important with pivotal trials, is to remove relatedness initially. They didn't report that in the 4 to 11 paper, but in the age 1 to 3, and that's defined as systemic allergic reactions or hypersensitivity or allergic reactions treated with epinephrine. It was 9% in active and 4.2% in placebo. Related ones, there's not a clear difference, but it was 1.6%. Remember, in the PITA trial, the toddler, it was 2% related. So it's actually it's very similar in that respect. And the epi use, there's a lot of noise here. So in the placebo, 3.4%, 6.8% in the 1 to 3, but also almost double that in the active group in both sides. So for whatever reason, epinephrine is used a lot more often than other safety things. And so we just need to keep that in mind. And that's something the FDA has to struggle with when deciding what to approve, for example. All right, so change gears again. What's going on in the new world with the next half an hour that we think in 2025 you can use with your shared decision making, or at least you should be aware of when you're making recommendations or deciding what to share with patients. because this is there's been an evolution of stuff here lately. This was uh, a real world study with PETA that was basically about 40, I think it was 45 allergists in different parts of the country that did a retrospective chart review of 118 patients. And this is just showing the demographics, basically. What I would point out is there, with 17.8% that were Afro-American and 10.2% Hispanic or Latino, this is more representative of the community demographic of peanut allergy than the clinical trial demographic, which tends to be really white and commercially insured. And you can see almost 20% were on Medicaid as well. So this is a misconception and illustrates another complexity in our health system, that just because something's quote expensive doesn't mean that that is a reason for poor access. I mean, there are these programs that companies have, and omalizumab, even in a more exaggerated way, is similar because it's even more expensive. But and we're not going to talk about price in this talk, but happy to do that as well. And then there was the clinical development program, or the pharmacovigilance program at Nestle, compiled all of the reports coming from the field. And as you may know, PETA, because of the 14% anaphylactic reaction rate, was under what they call a REM, so risk evaluation mitigation strategy, which is a something that I didn't even know what that meant when I joined Immune, but it's where there's more surveillance of the safety in a new drug that's on the market. And so every patient, every prescriber, and every healthcare facility has to attest ahead of time that they will report things like anaphylaxis. They also attest to making sure patients have epinephrine auto-injector and an anaphylaxis plan and things like that are kind of standard of care already. But but we think that the reports that come into pharmacovigilance are closer to reality than they would be without the REMS. And the good news is there was, in terms of epinephrine-treated reactions, it's 3.7% in the first 2,500 patients. And those that qualified as anaphylactic reactions, it was 1.8%. So no signal of a problem in the real world safety-wise based on anaphylaxis anyway. Okay, so let's get, sorry, my jumpy fingers here, too much coffee. So let's change gears again. Outmatched stage 2. So we're going back and forth, but this is relevant this year. We don't know a lot of detail because it hasn't been published, but it was presented, not just presented, but advertised by the Academy before the Academy meeting. And the objectives, which are all secondary objectives relative to the pivotal trial, which was stage 1, was to compare the ability to consume foods without dose-limiting symptoms. In other words, the amount of food you tolerate during double-blind food challenge after treatment with either the combination of omalizumab and OIT or continued omalizumab by itself without OIT. And then also look at safety. So and there were 117 patients that all received the stage 1 16 weeks, and then half of them went onto OIT with omalizumab for 2 months, and then continued OIT updosing after that. And the other half just stayed on omalizumab the whole time. So this was an adjuvant approach, which seemed reasonable when it was done. And the conclusion in February by the Academy's press release was that it's superior to oral immunotherapy, which is true in this study for sure, and it may be true. It depends how you define superior, but I would say it's at least misleading, and you should know the details behind that. This is just a look at the OIT protocol, and on the right you can see the updosing intervals of every 2 weeks, and there's protection by omalizumab until dose number 4, but then after that the omalizumab was withdrawn. So you're hoping that the desensitization process and hopefully early stages of immunomodulation have started to protect the patient. This is what we've seen in several other trials, but the reason I'm going over this is it wasn't apparent what was going on with this trial, because as you can see in the OIT treatment group, or the partially protected OIT treatment group versus monotherapy with omalizumab, the safety data endpoints were strikingly different. So serious adverse events, 31% in the OIT group. I've done a lot of clinical trials over the years. I've never seen that number. And so we need to have more detail. I think that FDA had a very conservative definition of SAE, but until we see it, we won't know. Whereas omalizumab placebo is 0. So it's interesting. I almost never see that either. Adverse events requiring epinephrine. Remember, there's noise. We'll give you 5%, maybe even 10%. And that's because placebo in here was 7%. Remember, the other trials, the Viaskin trials, the PETA trials, all had epi use in the 10% range. You're close to it in the placebo. Well, this was 37%. So I don't think that was, coincidence or noise. And then anaphylaxis was high as well. So something's going on here we don't really understand. Yeah. 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 So the question for those of you at home that couldn't hear is how do you monitor the patients to know how to do this, and is this safe? I mean, it depends who you talk to. Yeah. Yeah. Well, I think we'll be able to answer how to whether to come off omalizumab better when we see the publication. Bearing in mind that it's not a pivotal trial this stage, so we don't know, we won't be as confident about what it's telling us, but it's really important. I think that there's different camps, and I recognize bias in myself and try to be transparent about that. Well, in the OIT or card-carrying allergist immunotherapy camp, it's like, well, you went too fast. You should have, either done omalizumab longer or stop it, but back up a couple doses and go to a more conventional updosing after that. And maybe you would have avoided a lot of that adverse event profile that we saw in the first 6 months of PETA trials. So that's one camp. The other camp, and I'll channel Bob Wood, who's been the single champion of this entire thing, was that forget about oral immunotherapy. It's a bad treatment. Just do Xolair. It works for everybody. Yeah, it's tough. And to figure out if they're a responder, you have to do an oral challenge. Well, the answer, or the question was, do you do an oral challenge in clinical practice? And the answer is some people do, but the system can't support that. The infrastructure required to do that is. I'd agree. Yeah. Well, and you still have time to write an editorial, but there's not an article to write an editorial on yet, but hopefully that's being done. I was told by one of the authors that the data are confusing enough that other analyses need to happen. And this is NIAID. Okay, so this is, it's COFAR, it's not ITN, but it's still NIAID, and they care about mechanisms and being thorough. So I'm going to reserve judgment. But if anybody saw the presentation in San Diego and was confused, count me in with that group, because I just didn't know what was going on. Anyway, so let's change gear in the interest of time and talk about another really important study, which it depends whether you're an optimist or pessimist. This either is a wonderful, great thing for people, or it just makes it more complicated, depending on how you think about it, which is Scott Sisher from Mount Sinai has had a concept, which is, I think, now legitimized, which is a lot of patients don't have severe, quote, severe peanut allergy. In other words, they're high threshold reactors. And he did what's called his group did what's called the cafeteria study. So single center, pretty small study, but nevertheless very high quality the way they did it. And the entry criteria were not what you'd normally expect, which is that they're very sensitive and therefore they have peanut allergy and you've got an endpoint to start with. But actually that they're not very sensitive. So they only had to, they were excluded if they reacted to 4,000 milligrams, which is a meal of peanuts. So anything short of that with any reaction, they were included. And the median was being able to tolerate 2 peanuts, so 600 milligrams. And then they were randomized to either continued avoidance, which was what I've recommended for 20 years, or randomized to a very simple, cost-effective approach, which is peanut butter, starting with an eighth of a teaspoon, and sort of an OIT protocol done that way, for 8, and then followed by a maintenance of 1 teaspoon daily for 8 weeks. And then on the far right, you can see if they did a clinical trial. If they could tolerate 9,043 cumulative milligrams, they tolerated that, then they were a responder, and then they were said to go home and eat it if you want or not. And then after another 16 weeks, they said, we don't want you to eat peanut anymore for 2 months. And then they came back in for a final challenge. And we won't go through the whole study, but just to summarize, it went really well. So they 100% were desensitized and met that first outcome. The sustained unresponsiveness, in other words, so tolerated bigger than a meal of peanuts after 8 weeks of avoidance, was again that 67-68%. I mean, it's like each one of these trials seemed to be roughly 2/3 that do wonderfully. And then you can see and you saw the evidence of what you'd expect with immunotherapy at the bottom right, with the skin test wheel diameter decreasing. So I think even though it was small, it was pretty convincing that using peanut butter, which is like one of the least standardized ways you could treat somebody with a drug, it seemed to be quite safe, and have really good outcomes. They had no problems with systemic reactions. So I made this table to look at really good studies that we have and see, okay, is there some other way you can predict who these people are, or do you have to do an oral challenge? And I think my conclusion would be the latter, which you better do an oral challenge before you try to look at somebody and say you're a high threshold patient. And this is basically showing the age ranges of these different studies. The Poseidon and Palisade were the pivotal trials for PETA. The Epitope and PEPITES were the pivotal trials for Viaskin. And the reason I use those is because they have really good complete data on these patients because they're a pivotal trial. And you can see the eliciting doses, how they compare. The high threshold is by definition much higher. And then moderate to severe symptoms during the oral challenge. And it's not a trivial amount. The Poseidon epitope, the Poseidon trial, which is the toddler study for PETA, was 43%. In epitope, the Viaskin study was 94% had either moderate or severe symptoms. So it's not an innocuous undertaking to do oral challenges in your office. Although, honestly, a lot of the patients in those pivotal trials, you knew already that they were allergic. You just had to do the challenge. So we would call those high-risk challenges. So, but all this needs to be taken in context. The skin test wheel diameter is not a good discriminator. Peanut-specific IgE is a little better. Let me get rid of this thing. Oops, that's not good. All right. Whoops, that's bad. Here we go. But let's just say none of these biomarkers is adequate for defining them as high or low threshold. All right. So this is totally unrelated, but going to introduce the final topic today, which is how does it matter if you're using a standardized treating patient? In research, it does matter. The variation is the enemy of quality is a common phrase that started in manufacturing industry, but it also in science makes a difference. So you want to sort of know your variables and limit them when you're studying one thing and its change. in our clinic, it matters, we think, but it's not as 100% because you can't be 100% with things. You have to make judgments. However, just as a reminder, when I was first in practice, when this position statement was released, the Academy did endorse the use of standardized products. And this is why when you're doing SCIT and you have a grass allergic patient, you would choose standardized grass rather than some other northern grass. If you, most of us do that anyway, based on practice parameters. And that's because there's a 10 to 100, greater than 100-fold variation in the component allergen potency in general, with because these are naturally occurring, often grown in nature, like peanuts, or like eggs come out of a chicken. I mean, they're not exactly the same every time. Whereas standardized, when you've actually excluded those that are not within a certain range, they still vary 2 to 3-fold, but that's a much tighter. And there's manufacturer or product to product consistency, as well as lot to lot or batch to batch consistency within a given product. And in general, touted to be much safer to do with now, whether that's relevant for food, I'm not here to say yes or no, but I do think that we owe it to ourselves to consider this, that the discussion needs to be at a level that we're consciously choosing where the risks we're willing to take, or the patient is willing to take. So, and this is another table that is just showing or illustrating the different studies. So cafeteria study, Palisade, Poseidon, Outmatch, Impact, all these studies I've looked at before, except the second one, there's a retrospective chart review, which was Richard Wasserman's group a few years ago. 270 patients treated with peanut OIT. Their starting doses in the early, because remember, when you're starting in a sensitive patient, you need to start really low. And they started like 0.1 milligram. I mean, tiny, 0.01 milligram, a really tiny amount. And they use a suspension, mixing the peanut, PB2 peanut powder in distilled water and adding Kool-Aid for taste, and they've been doing that for years and report. In this trial, there were 23% that required epinephrine at some point during their treatment. And so it's, again, a bit above the noise that we're used to seeing with these things. With cafeteria, it was 0. I mean, they use peanut butter. So it's sort of, both were food grade peanut rather than pharmaceutical grade. With PETA, for the 4 to 17 and 1 to 3, you can see 14% systemic allergic reaction, all cause, 14% epi-use, all cause. And the PETA, only 2% related. It was 8% all cause, but it was the same as placebo, and 11% epi. Outmatch, and I put in red because it's just so shocking, these numbers. One thing to keep in mind that you may not have thought about, but Outmatch didn't use standardized peanut. They didn't use standardized food because they don't exist other than PETA, and they didn't use PETA. So, and halfway through the study, it was not, and this is in the New England Journal article, it was contaminated with mold, some of the lots. And so we don't know, I mean, whether there was some adverse event profile that was affected by that. But again, variation is the enemy of quality, and it just raises some concern. And the outcome for the OIT was really bad. It was only 19% that had success, mainly because of discontinuations and whatnot. It actually was made at the Stanford GMP lab. And then the food grade flour from Impact. Again, Impact had remember that was the one that showed remission in the really young toddlers. 22%. So again, a high number. So we don't know. Maybe that was related to allergen. Maybe not. Who knows? Without going into a lot of detail here in the last 10 minutes, I'll just say these are lots of PETA that were rejected because they didn't meet acceptability criteria. And the most common reason for rejection in the manufacturing process was failure to have consistent potency of RH1, 2, and 6, which was agreed on with the FDA as necessary to keep in that 2 to 3-fold range that we talked about. And it was the majority of lots. So this is a very impractical thing. If you're sourcing a bunch of peanut flour and you have to throw away more than half of it, then it becomes really expensive. So it's just, I don't know the answer here, but it's a fact that there's a difference there. And this just on the left shows what I just said, that the PETA lots were pretty consistent with the immunodominant components. And on the right is sort of an indirect marker of mold contamination. Aflatoxin is a carcinogen made by certain mold, and it's a common and expected contaminant of peanut crops. And you can see that the red dotted line, which you may not be able to see very well at 4 parts per billion, is the acceptability cutoff for a pharmaceutical grade. And so there were a bunch of lots that were rejected on that basis. And it seemed more of an issue as time passed. I won't spend a lot of time on this slide, but it's at the Academy, there was data presented showing the concentration of protein and the concentration of individual allergens in 12 peanut-containing products that are used for OIT. Not only 12 products, but 5 batches or 5 lots of each of those products. So they had people going all over the place buying different lots and then analyzing for protein content and potency. And you can see just in the on the y-axis is the number of milligrams per gram of whatever we're looking at. And on the x-axis are the individual products. The ranges, so when, for example, peanut butter in the US for RH1, this huge range is the range in those 5 products of that, or those 5 batches of that single product. So combined, you can see the variation in potency, the variation in protein content, and the variation lot to lot. And on the far left is the PETA. Thank God this turned out this way, because it's expensive. But the PETA is the narrow range on these, and similarly with the RH3, 6, and 8. So again, is this clinically relevant? You want to go back? Oh, yeah. So yeah, peanut containing puffs, which is on the far right. This is Bamba, okay? On the far right, really good for RH2. So it looks like PETA, okay? But for the others, not quite as good, but not bad. But then you get into what the next slide. I don't remember measuring allergen content and variability. Then the slide after this one is a unit to unit variability, which is another source of variation. Like if you pull one peanut M&M out of a bag and then pull another one out of the same bag, it's the same lot, it's the same product, different weights of stuff. So all the components are going to be different. Anyway, so it's again, a lot of sources of variation, which I'll show you a quick summary of in a minute. This is the last sort of data that was presented at the Academy, which is, remember I said that that the community that does unstandardized OIT, a common protocol used is to dissolve peanut, like PB2, in distilled water and add grape Kool-Aid because it tastes better, and then dilute it down to 0.01 milligrams or whatever. That has never been looked at. And so it was looked at by a guy named Martin Chapman in Charlottesville, Virginia, who is an allergen scientist that a lot of people have heard of. And he looked at all these things, and I'll just, and he also knows how to get peanut to go into solution, which is with PBS and other buffers that are really, it's not an easily solubilized protein. And so this is just showing the variation and the fact that Kool-Aid makes it even worse. So anyway, so without going into gory detail, just say there are product differences. There are lot differences in a given product. There are powder and flour measurement variability, like with a microspoon of 1/64th, and you're trying to get down to 0.1 milligrams or something. It's really hard to do that accurately, especially if the total protein of the source is high, because you're talking about a grain of sand or something to get, and it doesn't go into solution, as I just mentioned. So not that any of these is a deal breaker, but just I think we should be aware of the potential. Another thing that's important is that it's more relevant in low threshold patients earlier in their treatment, where they may, like at the Palisade study, which was 4 to 17 pivotal trial for PETA, the median highest tolerated dose at baseline was 1/30th of 1 peanut. So I think it's more important that they have an accurate dose while their threshold is still really low. And then during the updosing, as their threshold it starts exceeding the dose they're getting by more and more, it probably doesn't matter as much. But we don't know that either for sure. But I think it's probably a reasonable hypothesis. So in summary, um, defining likely responses to food allergy therapy is challenging, despite a lot of new stuff. And we have FDA-approved options now. Omalizumab is wonderful in that it has allergen agnostic and appears safe and effective in most patients. However, combining it with multi-food OIT had some safety outcomes that are concerning, and we need to know more. So hold that thought until we have the manuscript. And then with peanut OIT, bottom line, toddlers seem to respond better and in a safer, smoother ride than older children, and I think that validates a lot of anecdotal experience in this room. One small but very good quality single site study suggests that high threshold patients can be safely treated with grocery store peanut butter. And I think that, I'm happy with that. The product to product, lot to lot, and unit to unit variability in protein content and component allergen potency is actually, whether that's clinically relevant or not, it definitely discriminates standardized from non-standardized. And I think there are now data to substantiate that. But as I mentioned, the clinical relevance of this is unclear, especially for low threshold, but more likely in low threshold patients early in their treatment course. So I'll stop with 4 minutes to go and see if there are questions. Matt's had a few already, so